Okay, it is time for us to start. Welcome back, everybody, to the second week of the May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. We are very excited that you are joining us today. It's been a long program and it is quite intense, and we're very happy that you are coming back. And I hope that today's session will be very useful to you. The topic of this session is reproducible research in general and specific to, of course, mass spectrometry-based proteomics. Before we start, in case you're joining us for the first time, let me just go over the logistics. We have a chat box that you can use. We hope that you use it primarily for logistics issues, such as you cannot hear the presenter or the screen froze or something like that. And the panelists also use the chat box to broadcast messages to all the attendees. But for all the questions related to the actual topic of the presentation and related to the science, please use the Q&A box, and the presenters and the teaching assistants as possible will do their best to answer these questions either live or in writing. And as always, we hope that this session will be as interactive as possible. We welcome questions. Please do not be shy. And we hope that we will use this time to answer whatever questions related to your research that can arise. So without further ado, let me now introduce the first speaker of this session. It is my great pleasure to introduce Laurent Gatto. Laurent has been part of the May Institute for several years now, and we are absolutely delighted to have him back. Laurent has a PhD in evolutionary biology from the Free University of Brussels in Belgium. After his PhD, he spent many years working in Cambridge University in the UK, in the Center for Proteomics, where he worked on various aspects of development of methods for quantitative and spatial proteomics, and in particular doing this within the framework of R. Currently, Laurent is Associate Professor of Bioinformatics in the Université Catholique de Louvain in Belgium, where he heads the group focusing on computational biology and bioinformatics. Very importantly, Laurent is an accomplished educator and advocate for reproducible and open access research. So Laurent is a visiting scientist in the PRIDE team at EBI. He is an affiliated member of the Bioconductor project. He is a fellow of the Software Sustainability Institute, and he is an instructor in the software and data carpentry. We are very excited to have Laurent telling us today more about his work on reproducible mass spectrometry-based research. Thank you so very much, Laurent, for joining us.
Thank you very much for the invitation. So I'll get started right away. So if any of the attendees have questions, I'll do my best to try to answer them during the talk. And maybe if there are any more technical questions, then I'll carry on answering them after the talk and the demonstration. And so if you want to follow along the slides, get access to the code that I'll be writing, or get access to the lecture notes, you can access all these here on this URL. There's a little page about today's talk with links to all the details that you will be looking for. And so I want to start by thanking Olga and all the May Institute team for inviting me, of course. It's always great to be here, even if not physically this year. But I want to thank them for the work they do, for the work in sharing some very valuable tools, but also for the work in teaching the skills to use these tools. For a reason that I think will become clear later on, I think it's absolutely essential that as researcher, we are in a position to ask any questions we want to our data. And to be able to do that, we of course need tools to ask all the questions, but also the skills to be able to come up with these questions and then answer the questions. So let me start with a motivating example as to why reproducibility is important. And that example is the inverse problem. And inverse problems are extremely hard. So in this case, we're on the left-hand side for that score percentage that students get at an exam, for example, and then on the right-hand side, the grade. The forward problem is absolutely trivial. If I get a score of 68, my grade will be B. However, the inverse problem is extremely difficult. If I get a B, what was my original score? And we have a similar inverse problem in research. From the outside, I essentially get the grade, I get a paper in PDF. But what I'm actually interested in is the score that led to that grade. And here, the score is the data, is the code, the models, the results, the statistics, the software that are used to generate the paper at the end. And I'm sure some of you have already heard this or read this quote, but the paper in computational sciences, because I think it's not limited to computational sciences, is not the scholarship, right? It's merely the advertisement of that scholarship, right? The thing here on the right-hand side, this is not the actual scholarship. That's not what we are most interested in. The scholarship is everything that we have before that leads to the paper. That we should be able to access and understand as readers and researchers. And so why is reproducibility important? Well, I would say obviously for scientific reasons, right? Some people claim there is a reproducibility crisis, and we want to get access to everything that leads to the paper. But I think nowadays we also need to appreciate the importance of reproducibility for political reasons, right? In a time where the trust of the public in science, in experts, in data is not always as good as it should be, I think it's extremely important for us to provide as much clarity and reproducibility in what we do. But the point really that I want to make is that it's not only about reproducibility, or it's not about reproducibility for the sake of reproducibility. And it's not about open research just for the sake of open research, but really it's about reliable research, right? It's about trust. And openness and reproducibility are means that lead, or at least should lead, to greater trust in the research that we do and the research that we disseminate. So hopefully I have convinced you that working reproducibly is important, because that's how we can share more reliable science with our colleagues. But does it come at a cost? Well, probably yes. I mean, there is no such thing as a free lunch, right? Whatever we want to do, there is a cost. Obviously, in my opinion, that cost is worth. Generally, that cost is not financial, although sometimes it can be. It's mostly a time investment. And with the right tools and the right skills, among those that you are taught at the May Institute, these costs can be minimized. But I think an extremely important point, I think, for reproducibly, beyond the actual tools that you want to learn or use to work reproducibly, it's discipline. I think discipline is really what is needed to be able to work reproducibly. And no tool will allow us to work in a reproducible way if we don't put a minimum of discipline to it. But thinking again about whether it costs to work reproducibly, maybe actually it doesn't. Maybe the possible cost is more a matter of reallocating the time. And I really like this view of whether it takes more time to work reproducibly. The first one maybe represents a kind of a typical analysis pipeline or project pipeline, where we spend some time in gathering data, analyzing data, and then we wait quite a bit of time to get feedback from our peers, and then most of the time we need to redo some analyses, and then maybe we get additional feedback, and then yet redo some more analysis. And I think the beauty of working reproducibly is that actually we're going to reallocate the analysis time at the very beginning of the project. And so it might take a little bit more time, or even more, even if it's not a little bit of time, but even if it's taking substantially more time at the beginning, but that's time that is well invested, that we will kind of recover towards the end of the project. But so what do we mean by working reproducibly, or what do we mean by reproducibility? I think it's also interesting to think a little bit, what can we get using different tools or putting different time, different efforts into this reproducibility guidelines. And there is a kind of a classification of different levels of reproducibility. And the very first one would be aiming to be able to repeat an experiment, right? That would be to obtain the same tables, graph results, anything that is produced from an analysis using exactly the same setup, right? In the same lab on the same computer. Typically, that's if I want to repeat an analysis that I did yesterday or even a year ago. And even though that might sound trivial, it's not always the case. The second level would be to reproduce an experiment, not mine, and then again, trying to aim for the same tables, results, and so on, but this time in a different lab or in a different computer, using a very similar setup. So same type of software, reusing the same code. The third level would be the replication or replicate an experiment, and to obtain maybe not the same results, but at least similar enough results, and that's also something that would need to be defined, but then this time using a different setup. I might still be downloading the same data from public repositories. I might want to regenerate or re-simulate some of these results. And then I might, for example, re-implement the data analysis based on a description in the material method. And I'm sure that everybody that has read a scientific paper will know that that is certainly not an easy thing to do. And then finally, we have kind of the last level, which is kind of reusing information or knowledge from one experiment to reuse it, run a completely different experiment with the aim of confirming the results from scratch. And if we focus maybe a little more on the computational side, I think kind of this other tabular view is also really interesting, saying, kind of demonstrating or highlighting whether we use the same data, same code, and different code. And of course, if we use the same data and the same code, it's something that is relatively easy to do. If we use different code, it's not that easy at all. And of course, different data, different code becomes most of the time extremely challenging. So, all nice and good, but in my opinion, whether we want to repeat, reproduce, replicate, or even validate, what I think really is important is that what we want to do, what we should be aiming for, is to understand what has been done and what is happening. It's this understanding that is the most important, and that underlies the trust in the research we read or the research that we do. And so here, I'm not that much interested in kind of blindly rerunning something, simply to confirm that I get the same result without necessarily understanding it, which again, in some cases, is not easy at all. And I'll get back to that a little bit later. And also note that in some cases, things are not expected to reproduce, right? So if we use different versions of software, if the methods have changed, if bugs have fixed or the methods have evolved, have improved, we don't necessarily want to reproduce the exact same results that we obtained before. And so again, why is reproducibility important for myself as an individual researcher? And here I'll quote a talk that was given at the same May Institute in 2019 in Boston by a talk by Gabriel Becker. And where I think he made a really good point that reproducibility, whether computational or any, is not the point. We don't really care about reproducing something. We might actually be reproducing something that's wrong. but the goal is trust, verification, and guarantees. And so we have different level of trust depending on how much efforts we put in this level of replication that we want to attain. Is it simply trust in reporting? The results are accurately reported. If I say something, do I say the right thing? Or maybe I completely misunderstood my results. Trust in the implementation, right? For those that write code, we know that writing code is not something necessarily easy to do. So did I manage to actually write a code that does what I hope it should do? Statistical trust, right? Are the data and the methods that are used, are they appropriate, or are they still appropriate after some time? And then ultimately scientific trust, right? Are the results convincing? And do they support the claims that we are making? And so reproducibility and thus trust is really a scale from something here on the left that is absolutely not reproducible. It's essentially a black box. So we simply get a PDF of the paper and we need to take it for granted. Up to scientific trust. And this is a kind of a continuous scale. And depending what we are aiming for, we might try to move towards the right here as much as possible. And so big take-home messages: reproducibility isn't binary, it's a gradient, and is very often the result of the collaboration between different people that understand different aspects of the research, different aspects of the data. And reproducibility is certainly not something that is easy to achieve. But there are many more, I think, reasons to work reproducibly as a researcher. And these are very nicely illustrated by this paper by Laurent and Markovitz, which are 5 selfish reasons to work reproducibly. And I think that's probably the best reason why we should be aiming to work reproducibly. It's not for kind of ideological reasons. It's simply because it's good for us. It's reproducibility that will help us in achieving certain goals. And so here are these, very briefly, these 5 selfish reasons to work reproducibly. And the first one is that reproducibility helps to avoid disasters. And what is a disaster? Well, it's typically you do an analysis and you get it wrong. And then you want to write a paper or you want to present your results. Well, that's a disaster. We all do mistakes, but if we do mistakes, at least we should be able to understand our mistakes and fix them. And without any level of reproducibility, there is absolutely no way of doing that. And reproducibility also makes it easier to write papers, because by the time I want to write, describe my results, write my conclusions, there might have been several months since I actually did the analysis. And so I don't need to kind of guesstimate or guess what I have done weeks or months ago. I can actually check it directly in my code and repeat it if I need it. Or if I don't understand figures, I can go back and understand the code that produced these figures to make absolutely sure that I know what they are supposed to demonstrate or to explain. And that not only holds for myself as an author, but it also holds for the reviewers. And if I can demonstrate to the reviewers that I provide not only the final PDF, but the data, the code, and everything that led to these results, that's a very good way to convince the reviewers that my results make sense. And reproducibility also enables continuity in our work. And that's extremely important, of course. We don't want to kind of start from scratch every time we have a new data set to analyze. We want to be able to reuse our previous work. And that's one of the things that reproducibility allows us to do. And finally, reproducibility helps to build reputation, right? Coming back to the point of trust, generally we will favor the results, the papers from authors that are known for working reproducibly. Authors that provide the data, authors that provide the code. So really, working reproducibly is an excellent investment of time. So what are the tools to work reproducibly? So I'll demonstrate some easy ways to work reproducibly using R and bioconductor packages, and I will assume that many of you are familiar or even experts with R. And my preferred way to write reproducibly is to write scripts or RMD files to keep track of what I do and be able to easily rerun the script. And the packages that I will demonstrate are those from the R for mass spectrometry initiative, which I'll describe a little more on the next slide. And so this is a kind of a relatively, I don't know, maybe poor man's way to work reproducibly, but it works for me. But it's possible, of course, to set up a completely pre-configured environment. And if you are familiar with Docker containers or Amazon machines instances, or I use Ranku, for example, to teach, that's also really great, because instead of simply providing a piece of code and instructions of how to install the software or how to run the code, it's possible to provide a completely pre-configured environment to fellow researchers. And in some cases, that's extremely valuable, right? If you run a 2 hours workshop, you can't spend time. What is bioconductor? I'll explain in a second, right? If you have a 2-hour workshop, you can't spend time on installation problems, and in that case, it's really great to provide something that is already nicely packaged. But I think as researchers, investing a little bit of time to install our software is actually something really useful. And so R is a language and working environment for statistical computing. And one of the great features of R is that we can install additional packages that provide specific functionality. And bioconductor is a project that provides packages that are dedicated to the analysis and comprehension of high throughput biological data. Bioconductor was actually initiated by one of the 2 original authors of R, Robert Gentleman, and it's a set of packages, packages that work coherently together to analyze high throughput biology data. And so the couple of packages that I will highlight today are some of the packages from the R for mass spectrometry initiative. And many of these packages are already available through bioconductor. So the links above will provide you much more information about the R for mass spectrometry initiative. But the aim of this initiative is to provide efficient, thoroughly documented, tested, and flexible R software for the analysis and interpretation of high throughput mass spectrometry data, whether it's applied to proteomics or metabolomics. And the project formalized the long-time collaborative developments and efforts from 3 people, the kind of co-developers of the initiative, which are myself, Sebastian Gibb, and Johannes Reiner. And so we have been working together for many, many years now. And then I think it was in 2018, 19, we realized that it would be really great to be able to start from scratch and do some of the things we did, but do them in a kind of better way. And so there is kind of one slide of theory or background information that I need to explain before being able to dive into the code. And that's how do we see, or how do we think of mass spectrometry data when we work with the R for mass spectrometry infrastructure. And that raw data is stored and can be manipulated using the Spectra package that we'll see a little bit later. And so when you think about mass spectrometry data, we essentially think of 2 parts. The first one here on the left is a collection of spectra. Right, we have amyloid Z values and intensities. That's the raw data. And then each of these spectra have an annotation. And all these annotations are stored into a big table. And so each row of that table provides the mass level, the spectrum index, the acquisition number, a mass level, polarity, precursor M over Z, and so on and so forth for each of these spectra. Right? That's really the only thing that you need to know to be able to get started. And these different parts of a mass spectrometry data set can be stored in different modes, right? In different ways. They can be stored in memory, they can be stored on disk, using different formats. And that's something that we call backends. And then towards the end, hopefully I'll get time, I'll briefly mention backends. But that is completely transparent to the users, right? So later on, I'll be manipulating some data, and so think of these data as a collection of spectra and a big table that annotates all these spectra. And so I'll be focusing on the raw data infrastructure centered around that spectra package. So how to handle raw mass spectrometry data. If you're interested in quantitative data, that's something I will not be talking about today because I won't have time. Have a look at the Q features package. Okay, so let me remove this and bring this other window to the front. And so let me get started by simply copy and copy and pasting this piece of code that for those that had a look is also available on the conference page. These are the lines of code that allow you to install the very latest versions of all the packages that are needed to repeat or reproduce the code that I'm going to show. Now, in a couple of weeks, once the latest version of Bioconductor is released, you won't need to install these kind of development versions, but some of the features that I want to demonstrate are not available in the kind of release, the stable version of all these packages of Bioconductor. And so the first thing I would like to do is to download some data. And to do that, I'm going to use, split my screen here. I'm going to use, oh, no, I don't want, I don't see, oh. So I need to change it a little bit because with the Zoom screen, I don't see all my screen.
Laurent, and there is a question. Can you tell everyone what R coding environment you use? Because we have talked about RStudio in the previous days, so this is something else.
Absolutely, yes. I use Emacs for all the work I do, including R coding, but this would work exactly in the same way with RStudio, right? So it's simply a window here on the right-hand side where I have some code, and I'm going to execute that code on the left-hand side. Okay, so it's really the equivalent of this. It's really the equivalent of RStudio. And so the first package I would like to use here is a package called RPX that will allow me to access data from the Proteome Exchange repository. And I'm going first to specify the data set that I'm interested in using the pxData function. And I'm going to say I'm interested in a specific experiment. And that specific experiment, I'm going to copy and paste the information about that paper here. And with this, we don't need it anymore. So we're going to take a subset of the data that was published earlier this year as part of a paper called Raw Beams, a simple vendor-independent raw data quality control tool in Journal of Proteomics. And the identification number of that data set is pxd 022816. Right? That's kind of documented in the paper, and that's what I'm going to use. And I see do you think there is an obligation to provide your raw data and code? Well, it's not for me to decide. If you want to publish your research in a journal, it would be for the journal to decide. And most of the journal will expect you to indeed publish your data, most certainly. I'm not sure about the code, but reviewers might ask it. But personally, I think, yes, there should be an obligation to publish data and code, assuming, of course, there is no private patient information. In that case, that data would need to be anonymized, right? But except for that reason, I definitely think that it's an absolute obligation to publish these. So back to my code. Let me increase the font here on the left-hand side. I'm going to create a variable, an object called px, that kind of encapsulate all the information that is available in that project. And I can use the function px files to get a listing of all the files that are available. And we can see here that we have a set of what I assume is 10 fractions. So this data has been generated on the Q executive, if I remember well. And for each of these fractions, we have the raw data file, the binary file as was produced by the instrument, which is shown here. And that file converted to MZML, and that's what we're going to use. And then we also have the identification data as an MZID file. So let me store this into a variable called FLS that contains the name of all these files. And so what I'm going to do now is that I'm going to download these files from Proteome Exchange. And I'm going to download the MZMLs. And these are those that have the extension MZML. And so the pxcat function is a function that's going to go and check in the pxd and so on repository, or the Proteome Exchange repository for that specific project. And here I say that I want to have the first 3 MZML files. So that is fraction 1, fraction 2, and fraction 3. And I'm going to do the same thing here for the MZID. Oops. Sorry. I don't want to reinstall all the packages. So let me come here. I'm going to now download the 3 MZID files, right? Now this was extremely quick because the files didn't need to be downloaded. They were simply loaded from cache. So the RPX package tries to be clever and will not download different data sets multiple times. When the data is already available, has already been downloaded, it will simply be reloaded from cache. But so what I have here are 3 MZML files and 3 MZID files that I will be using now. But I think one way to facilitate work reproducibly is to try to store all the data together in one place or under one variable. And so when we do experiments, or when we do mass spectrometry and proteomics, we kind of think of these data as part of an experiment. And so what I'm going to do, so I'm always going to load my packages at the very top, and that the code that I'm writing now will be shared with you. I'll push it so you have it to the link through the link that is available on the talk web page. I'm going to use another package called MS Experiment. So let me load it. That I want to use to kind of annotate and combine all the information that is part of this analysis and all the information, all the data that constitutes my experiment. And so I'm going to create an object MS Experiment. I'm going to call it MSE here. MSE, and as you can see on the left-hand side, it says object of class MS Experiment. It's an empty experiment. I don't have anything yet in that experiment, but what I'm going to do is that I'm going to slowly populate it to have a coherent object, computational objects that store all the bits of that experiment that I deem important. And so the first thing that I would like now to add to this experiment are the files that I downloaded. And in this case, I downloaded these files from Proteome Exchange, but these could just as well be files that were generated in my app and that are already available on my disk. And so I'm going to say MS experiment files to kind of store these in a coherent way as MS experiment files. And I'm going to say that my, well, MZML files will contain the files that are stored in my MZML variable here. And I'm also going to say that I also want to have MZID files. Let me check, how did I call it? It doesn't matter, you can call these the way you want. And here, oops, again. So I will going to comment this, otherwise it's going to try regularly to update my packages. So this is my way to say, well, now this experiment, or for this experiment, I have a set of files, and they are stored here as MZ. I wanted to call them MZMLs and MZIDs. And these files, I now want to add. So if we look back at this MSE, if I ask, well, are there already any experiment files in this MSE? It will tell me no, there aren't any. So what I'm going to do is that I'm going to assign these MS experiment files to this experiment object. And now, before it only said that I had an empty MS experiment object, and now it tells me that as part of this experiment, I have a set of files, MZML files and MZID files. And so that's a start, right? Knowing which files belong to an experiment, again, sounds trivial, but I'm sure many of you have already had a situation where you couldn't find the files that were part of a data set. Well, that's one way to keep everything together.
Laurent, there's a couple of questions that may be good to answer now.
How would the beginner know what utilities are available? Yes. So let me see. For mass spectrometry, there is a web page for the project, and that's of course only one set of tools, right? There would be many other sets of tools, with here a list of packages that you might want to use. And for example, the MS experiment, you can click here, and this leads you to a rather detailed documentation with many more details than what we will see today that explains what you can do with these experiments. And there are some kind of non-trivial things that you can do with these experiments. And then there is also another tutorial here that provides, again, kind of use cases of what kind of things you can do with all these packages, and depending on what you want to do, which package to use. So one of the efforts we do as part of our mass spectrometer is really to provide up-to-date documentation. I don't mean to say that suddenly with all this documentation it becomes straightforward for a beginner to start analyzing their data with these packages. There is a learning curve, of course, but I guess that's the price to pay. But the documentation to make that as easy as possible is available. So what do you think the TAC replicate using mass spec? And by TAC replicate, do you mean technical replicates? And regarding all the links that I have shown are already available. So if you go on the Google Doc, or the link on the conference page, you will get access. You have access to all the links and all the slides and all the code that I'm showing. And so, regarding the previous question, I'm not sure what you meant, but if you meant technical replicates, I certainly believe that biological replicates are much more interesting than technical replicates, but that's more of a experimental design question, I think. And so in px get here, to answer the other question, px is the object that represents this proteome exchange experiment. And here px FLS contains the list. We can look this here. FLS contains the list of all the files that are interested in. And here I want to get not all the files, but I'm only going to look at the first 3 files. Yeah, and so technical replicates. I don't think technical replicates are the best investment of our time and money. I would certainly rather suggest to do biological replicates, as long, of course, as the technical aspects are handled, right? This, of course, assumes that the kind of running the experiment is something that is in place. And so coming back to my MSE object, well, currently I have an experiment that contains file. In a minute, we're going to add raw data, but I might want to add additional information, any information I'm interested in. Right? So for example, if I look at the metadata, well, metadata is empty, it's an empty list, but I could say, pxt, and of course, something that would be interesting would be to store here the proteome exchange identifier of that experiment, and then maybe the PubMed ID. Let me store this too here. Here, let's say PubMed ID. And so I could kind of add these things to my MSE object. And now if I say metadata MSE, it tells me that I have a pxt element and a PubMed ID element, right? So I could really add any metadata, any information I'm interested in here to very simple elements, but these could be much more complex, and some of these are described in the vignette. So, very good question. So these packages only focus on data that was exported into one of the open formats, such as MZML, MZXML, and so on. Right? They do not read raw or binary files, but as long as you can convert to MZML, it will work. Yes, and so if you get this error, I assume that pxFLS, that variable, was not created in your case. I'm not expecting people to type along with me. You will have all the code if you want to kind of replicate or repeat what I did afterwards. So I think it would be probably a bit of a challenge to try to repeat everything I did. Okay, let's go to the next step of our data and load the raw data. And to load the raw data, I will use another package called Spectra. And I'm going to load that package here at the top. So here in my environment chunk, I will, every time I need a new package, I will load that package there. So it's easy for me later on when I want to understand what my script did. It's easy for me to kind of go back and check what were all the packages that I needed. Okay, and so once I load the Spectra package, I have access to the new function called Spectra that will create such an object that contains all the raw data. And which files do I need to generate such an object? Well, the MZML files. And where do I have these MZML files? Well, in my experiment, I can extract the experiment files of MSE, okay? And that's a list with 2 elements. And among that list, those that I'm interested in are those that are called MZMLs. So these are the 3 files that contain my raw data. And so given that I'm working in the frame of this experiment, I'm not going to retype file names or get them somewhere else, I would immediately use the file names that already exist in my experiment. So all the data can simply be downloaded from Protein Exchange. You're absolutely right. You can go on an FTP site and then download the files manually one by one. You're absolutely right. The advantage of doing this here is twofold. Well, the first one is that I see exactly which files I download. Okay, so there is a script, a piece of code that demonstrates which files I downloaded, which is something that you could do with discipline and write it down in your lab book, right? That would be just as good. The second advantage is that this will automatically cache your data. So if I download it once, it will not download it again if I need them, in another directory. But a lot of the things that I'm presenting here could be done in different ways, for instance, manually, and that's perfectly fine, as long as you have the discipline to work reproducibly, without using some of the tools that I present that help you work more reproducibly. So I'm going to create a new Spectra object with the files that I have already stored as part of my experiment. And I'm going to call this object SP. So here we go, here on the left-hand side. SP now contains a raw data object that contains 87,647 spectra, right? All the spectra that were present in these 3 files here that we downloaded from Protein Exchange. And now it's kind of useful to remember that slide that I showed where when I said, well, a spectrum, an MS experiment is composed of 2 things. One big table that contains all the annotation. And what is this annotation? Well, that's something we can get with the function spectra variables. If I say spectra variables SP, it lists a vector of 34 elements. And these elements are the annotation that we have for each of these 87,000 spectra. So we have the MS level, RT for retention time, acquisition number, polarity, whether that's centroid or not, and so on. So that's all the information that is contained in that table. It's a table with 87,000 rows and 34 columns. And so spectra variables allows me to get the name of these variables, these annotations. Spectra data allows to actually get that big table. Now, it's too big to be shown, so we only see the first row, but spectra data allows me to get that big table with the annotation of all my spectra. So what kind of results am I trying to reproduce? I'm not trying to reproduce anything specific from that data. I'm going to show how to load data, how to do some quality controls and visualization of the data. I'm not trying to replicate anything specific. But the work that I'm doing now, you should be able to repeat it by installing the package. So I was saying, remember big table, spectra variables, and spectra data. That's how we can access the information in that big table with the annotations. And then there was a second piece of information in a raw data experiment, and that was the M over Z and intensities. And that's something that we can access with peaks data. So if I say peaks data, before I get to that, so SP contains my full experiment of length, 87,000, and I can very easily take subsets of this experiment. So for example, if I simply say 1 to 2 here, SP 1 to 2 behaves like a list, and we'll create a new experiment with now only 2 spectra. And so if I want to access the so-called raw data, I can do that with the peaks data function. Okay. And the peaks data will actually go and collect the MZ, LZ values and intensities. So let's have a look at the kind of M over Z and intensities of the first spectrum. Here there are, right? So that's the first spectrum in that data set. So we can go and access the pieces of information that we are interested in. Now we can also visualize these. So instead of looking at the matrix here, maybe it's easier for me to say, well, plot spectra. I don't want to plot the full experiment, but only the first one. And it appeared here at the back. Here we go. So that's the visualization of the first spectrum. And here on the left-hand side, you have the actual data. Right. That was, where is it? That is a spectrum MS level 1, retention type 02. So that's the very first spectrum that we have in this data set. If I type any number here, and before I tried with 23822, and if I rerun this, this is an MS2 spectrum. You can see here MS2 at this retention time. So it's relatively easy to extract, and if we want to visualize these data. Well, let me now load another package very briefly. That is called SpectraVis. And with SpectraVis, what we can do is the following. We can use a function called plotly spectra. And that one will open, and it opens here, kind of an interactive way to visualize these data. So that's the same spectrum, still spectrum number 1 shown here, but it provides some flexibility. If that's what you need to do, you can zoom in into specific regions of your spectrum. Right, so once you have the data, you can easily visualize it either using static or interactive visualizations. But so we have all that data, so let's try to explore it a little bit. And let's try to check, yes, if you use RStudio, it will still be interactive. This doesn't use any custom infrastructure. It uses the plotly package that also works with RStudio. And for any errors, I will look at these later on and try to debug and help you with the installations of some of the packages. So how do you get the M over Z and the charge? Well, that's an example that we can have. The charge of the precursor, well, we could say precursor charge, sorry. Charge here is P. And this typo precursor charge, this would kind of get us the precursor, the charge of all the precursors. Of course, if we have an MS1 scan, there is no precursor, so we have an A. And so, for example, here we can see that among all these spectra, in this case, the MS2 spectra, we have close to 46,000 spectra that have a precursor charge, or MS2 spectra that have a precursor charge of 2, and then 20 or so, 30,000 of 3, and so on. So that would be precursor charge. And then if you wanted to have the M over Z values, well, you can access these with the peaks data function. So same thing for the intensities. But so let's have a little look. So SP is this raw data object, and there is the data origin variable that tells us where do these spectra come from. And so if we look at the table of this, we see that we have more or less the same number of spectra in each file. Right? 29,000 in the first file, 28,400 in the second, and 29,300 in the third. Okay, and then something else that I find actually quite interesting is to say, well, here we have the MS levels of all these. Let me show this. Okay, of all these data. Actually, we can maybe table of this. So we have 19,000 MS1 level scans and 68,000 MS2 level scans. And we can also check whether these are centroided. Put the centroided here. If I only look at the 6 first ones, they are all centroided. And something that I actually find really interesting is to have a table of MS level by centroided. And in this case, I realized that all my MS1 spectra is centroided and all my MS2 spectra is centroided. But very often you might have MS1 spectra that are in profile mode and MS2 spectra that are centroided. And if you have MS3, you could also kind of tell which ones are centroided and which ones are not, which is something that might be interesting for downstream analysis. But we have all this raw data, so let's try to kind of make use of these and do some visualization. And so I'm going to go back at my very first part here and load 3 other packages: ggplot2 for visualization, tidyr for some data manipulation, and then magrittr to use the pi operator. I'm going to load these 3 packages here, and let's do some visualization. And the first visualization that I would like to do is to visualize the chromatograms. So let me add this here, the chromatograms. So chromatograms only need MS1 data. So I'm going to use the filter MS level function on my SP object to only retain MS level 1 spectrum. And so we know here that we have 19,607 MS1 spectrum. And then I'm going to extract this metadata, also it's this big annotation table with the spectra data function that we have seen before. I'm going to convert it to a table, and then I'm going to use ggplot to visualize the total ion current along the y-axis along the retention time. And I want to separate this based on the data origin, which is the file where these are produced by. And so here is that figure. Let me make it wide. Right, so we have extracted the information that we see here on this figure, the retention times along the x-axis and the total ion current along the y-axis. And here we can visualize the total ion chromatograms of these 3 row experiments. And we can confirm that they are actually quite similar, some difference at the beginning, but these look like very good chromatograms. Something is maybe a little bit surprising here for the second file, in green here, that's the, oh no, sorry, the third file. Somehow, apparently, there is a short time during, what is this, 1,800, 1,900 seconds, where suddenly we don't apparently have any MS1 scats that were acquired. No idea whether this is something we should be worried about or not, but at least it allows us to easily and quickly produce these chromatograms, which are a really good way to kind of QC your data. Okay. And then something else that I find really interesting when it comes to quality control. And I'm going to run this because it will take a little bit of time, and you can get the details of the code here. I'll leave this command in the code because it's not in the package yet. So we are going to only extract here the first file. Right. Filter data origin will tell that, among all the, 87,000 spectra, only keep those that belong to the first file. And then I'm going to call the compute MZ deltas function. And so what this function is going to do, it's going to first only extract the MS2 spectra. What is the environment that I use? So I use, let me type it at the very beginning. I use the Emacs software, and the kind of package that allows me to manipulate this R code and run it is called ESS. Okay, but just for your information, our studio is really great. And so if you are not already familiar with Emacs, I would not advise you to get into Emacs just for the sake of using or using R. Our studio is really a great piece of software. So by all means, use it. And so, so this MZ delta or compute MZ delta is going to extract all the MS2 spectra. And an MS2 spectrum is supposed to represent some information about the identification of these peptides. This is a proteomics experiment. And the peaks that we see in the MS2 spectrum are supposed to reflect peptides, or the difference between all these peaks is supposed to reflect the masses of different peptides. And so this function is going to take all the MS2 spectra, only consider the highest MS2, the highest peaks in these MS2 spectra, all the fragment peaks, and then it's going to calculate the distance between all these peaks. And if what I have analyzed here, or if the data that I have downloaded actually analyze peptides, and we know that's the case, well, then I expect that all these distances, these differences between fragment peaks in my MS2 spectra correspond to the masses of amino acids. And so this function is going to calculate all these distances between all the fragment peaks of all the MS2 spectra. Well, actually, maybe it's not all, it's only a kind of certain percentage, or only the fragment peaks that are in the top quintile or something like that. And so it's going to do that. And it looks like it's taking a lot of time, and I think that's because Zoom is taking quite a few resources. Let's see. Okay. Yeah. Okay, let's give it a little bit more time. Here we go. And so if I look at D1 plus D1 here, it's a list. And you see, this is a list with all the deltas between the fragment peaks of my MS2 spectrum. And now if I visualize this, where is my figure? Where has it appeared? It has appeared here. I obtain this histogram here. Not sure, can you see my histogram?
Yes, yes, we can.
Yep. Good. Well, along this histogram, I am highlighting with kind of vertical lines the masses of my 20 amino acids. And indeed, I observe that I have an increase of these MZ deltas in this data. So that is typically the kind of histogram of mass delta distributions that I want to see. I want to see an increment in mass deltas corresponding to peptides. As many, or as everybody have already had experiments that completely failed, and then you would get essentially a flat histogram, which would be a really good indication that even if you run a search engine, but maybe your parameters were not optimal, this will give you a kind of an unsupervised quality control of your MS state. And so will this work for modified peptides? Yes, it will work. You might want to also highlight the masses of your modified peptides, but this should also work for modified peptides. Yes, it's completely agnostic of the technology that you used. Okay. And so that's another type of quality control that I find really useful and that I tend to do, especially when I don't quite get the number of identifications that I'm supposed to get. It's always good to try to have a, let's say, search engine independent quality control. But so I have here my SP object, of course, and it still kind of lives outside my experiment. And so if I want to add it to my experiment, I can say spectra MSE and say SP. So now I'm going to kind of add that spectrum object to my experiment. And now if I simply print the name of my experiment, it tells me, well, I still have the files that I had before, but now this experiment also contains that many MS1 spectra and that many MS2 spectra. And I'm not going to do it now because it's not very interesting in terms of demonstration, but I could save this object, close R, reopen R, and simply load that object, and then I would get the raw data, all the files, and any metadata that I have added. So let's have a quick look now at the identification data that we had. And for that, I'm going to use a package called PSM. And then I'm going to load that package, but I prefer to put all my package loading at the top. And now I'm going to load these identification files. Where can I get these identification files? Well, with experiment files. Okay, here they are. And then $mzids. Here are the 3 identification files that I downloaded. So I'm going to load these into with a function called PSM into a variable that I call ID. And it's kind of shown here. This is an object PSM that contains information from MZID files, and it contains 74,750 PSMs. And I have 31 variables or columns that I can look at here, names, ID. So I have here these 31 annotations for all my PSMs. And so I can extract these, and I can manipulate these. And one visualization that I'm going to do is the following one. So this ID variable, it says it's an object, it's a PSM, but really it's only a kind of a glorified table. And I can very easily convert it into a tibble. If you are familiar with tibbles or data frames, and here it is. So simply by saying as tibble, I convert it into a tibble, and then I'm going to feed that tibble into ggplot2 to produce a visualization. Before I say I have a question here. Yes, that is exactly what I was looking for. These MZ deltas should roughly correspond to the masses of amino acids. Yes. Without considering any information about the search engine, I kind of look at the raw data in an unsupervised way, and I just want to make sure that these deltas correspond to amino acids. And so with this piece of code, I'm going to plot the metamorphosis score, actually density of these scores, and then I'm going to color code these densities based on whether that PSM comes from the decoy database or the forward database. And this produces this figure here. And so I split these figures based on the spectrum file, the raw data file. And so that's, I think, also the kind of visualizations that are always extremely useful, not just look at the number of identifications, but here I can clearly see that those that are decoy peptides, so in blue, tend to have much lower scores than those that are in red here, which are non-decoy peptides, right? So I want to see a distribution shifted towards higher scores for my non-decoy peptides. And this is something that I did for the metamorphosis scores, but I can do exactly the same thing for the, what did I use here? The PSM level Q values, right? And so we see that we have much smaller Q values here for the non-decoy peptides. But note here that actually these identifications that I downloaded through these MZID files have already been filtered. As you can see here, we have Q values that are smaller than 0.01. So this is already our 1% data set that contains the decoy and forward peptides, but I see a clear enrichment here for non-decoy peptides. So good quality identification data. Now, of course, we could kind of look at these identification data in great detail, but in this case, in the interest of time, I could simply filter these PSMs, remove all the decoy hits, remove any PSMs that have a rank greater than 1, only get rid of the non-proteotypic peptides, and this would leave me with 3,700 high-quality PSMs. How do you make it work for TMT labeled peptides? Yeah, no, I don't think that's a problem with TMT peptides because you don't have all the single amino acids that are labeled. So the MZ delta histograms also works for kind of labeled peptides. So here I have all my PSMs, but I can use the filtered PSM to, and let me overwrite this object here, to now get a reduced set of 36 or 37,000 high confidence PSMs. Now I have my PSMs, I have my raw data, but what I really want to do is to combine these, to join the raw data and join the identification data. And for this I need, yes, so the filtering, so this is kind of one function that does lots of filtering together, but in the interest of time, I quickly use it like this. But yes, you can parameterize the type of filtering that you want to do. Is there a Google group? There is no Google group, but we have, we answer to questions on GitHub, or we also use the, you're also free to ask questions on the bioconductor support forum. And this is described, where to ask questions is described on the respective web pages. And so what I really want to do before, because I see the time is running, is to combine these raw data and these IDs, these identification data. And so to be able to combine these, we need to have a shared variable from the raw data. And you remember, if I look at the spectra variables, these are those that I have, right, in my raw data. And then if I look at, what was it, names, ID, these are the variables that I have for my identification. And I somehow need to be able to match these. And that's not difficult to do, given that I have file names and was it spectrum file and spectrum file, right? So data storage here and spectrum file here represent the same information. The raw data file, here spectrum file is the raw data file from which the identification comes from. And the second information is the spectrum ID. So where is it? I can't quite spot it. So I have spectrum ID here for my identification data. And I also have a spectrum ID for my raw data. Where is it? Spectrum ID. Oh, here it is. The difference, and that's the nature of the data. I have a lowercase d here and a capital D. No, I lost again. A capital D on the other side. But I have all the information that I need to be able to precisely match spectra in specific files between these two data types. And so I'm going to do that. And you will have the code, so you can look into details by creating what I call a primary key. And the primary key here is composed of the file name and then the scan number, right? So I know that the 14th element in my raw data file is scan number 14 out of this first file. If I look at the end, tail of this, I know that my last element is spectrum 28,740 from my third file, right? So now this P key, that is a concatenation of the file names and the spectrum identifier, uniquely identifies all the spectra in my raw data object. And so I'm going to do exactly the same thing for my identification data. So here's the code. And again, hat ID $primary key again allows me to say, well, my first PSM, it comes from this file and this spectrum identifier. And so that's a way to be able to kind of uniquely make links between raw data and identification. Now, again, to simplify my task here, if I look at these, let me look at the first ones. Here they are. Look, actually, I have two PSMs for scan 15857 in my first file, and that's not really unexpected. The fact that a scan can match multiple PSMs. And just to keep things again simple here, because we don't have the time, I'm going to get rid of any scans that have multiple matches. There are ways around this, and it would be really interesting to look at these data into detail. But here, for the sake of time, I'm simply going to kind of cheat and simply take the those that match uniquely. And so what I'm now going to do is that I'm going to join spectral data, join information from my raw data, and join information from my filtered data. And both can be uniquely matched using the P key, primary key. So let me overwrite my object SP, and now check back into the spectra variables. And now I see that for each spectrum, I have my original information, but I also have information about the identification. And so here I literally joined the information from my raw data. And I've done it all manually. I mean, that's kind of the point here, to explore, to become very familiar with my data, to join these pieces of information. And so, yeah, there is an error, but I'll look at these later on. So why do you get two entries for scan number? Because I have a single scan that was produced by my mass spec, but I had two possible matches for this one. For example, if there is some uncertainty, there is not always a kind of unique matches. This happens. Actually, I didn't. It was for a minority, a small minority of cases, right? It happened that I had the case here at the very beginning and the second one here, but it was a minority of situations. Could you add the code later? I can add the code later, but I think the best thing to do is to look at here in this tutorial, right, and the links are in the notes. And here you have kind of documentation as to how to handle identification data, lots of examples. So all the information is available. And so now in this SP object, I have joined raw data and identification data, but I have done this in my SP variable, not in my experiment. So what I'm going to do here, spectra, is to overwrite, kind of the part of my experiment that contains the spectra. And so now in my experiment, I have the links to the files that are used and my raw data, but this time this raw data also contains the identification. So do you get two entries because the search allowed ranks 1 and 2? This could be a reason, but here the filtering that I did, where was it? Here, the filter PSMs actually only keeps only the first ranks. So you're absolutely right, Susanna, but in this case, it's for another reason. And so, so here we go. So I have this experiment, and I'll just I'll tell you to, if you're interested in this, look at the vignettes about experiments and raw data. There's of course so the MS experiment package and the spectra package. There are many, many more things that can be done if you're interested in. But what I'm going to do now is that, let me copy this over here. Right, I have, what is it, 87,000 spectra. Not all of these are equally interesting. So what I'm going to do here is that I'm going to say, well, now that I have joined my identification data and my raw data. Just do this, right? I have all the database accession numbers for the spectra in my raw data. So for example, I can see that this identification number contains 2 spectra. So this protein was identified by 2 spectra. This other protein was identified by 4 spectra. So what I'm going to do now here is that I'm going to create a subset of my raw data that only contains 13 spectra, with all the spectra that were matched to peptides belonging to that protein. Okay. So SP is my raw data object, contains 87,000 spectra. Now SP underscore 043175 only contains 17 spectra for that protein of interest. And very often, once you get to the very end of your quantitative analysis, your statistical analysis, you get some really interesting hits. It's often really good to go back at raw data and somehow make sure that your raw data, whether it's in terms of identification or quantitation, actually supports your final conclusions. And what I'm going to do here is that it's a little bit technical, but I'm going to kind of change one backend to another one. So everything I have done was actually really fast. Reading these 87,000 spectra into R was really fast. And that's because the data was always stored on disk. So I kind of saved a lot of time by not loading the data into memory, but keeping it on disk and only access it when it was really necessary. But now, given that I have a really small data set, I'm going to say load that full data set into memory, like change the backend, so that now these 13 spectra, a tiny data set, are readily available in memory, and that can be very fast to manipulate. And why do I want to manipulate these very quickly? Because I will want to do some clustering. With the compare spectra function is going to take all pairwise comparison between these spectra. And I'm simply going to give them some names to make it kind of easier to look at. And it's going to, I can now easily create this heat map where each cell here corresponds to the similarity between these two these 13 MSMS spectra. Right, so the similarity is 1 along the diagonal, because here I compare one spectrum against itself. And then here I have one cluster of spectra that are very similar. They have distance between 1 and, 0.6, also 0.6, 0.7. But in here, I see that I have 2 subclusters. And they all have the sequence DLP something. So they have all been assigned to the same peptide. But I do see that within this peptide, I have 2 subclusters. And we'll have a quick look later on what this means. And here I have another cluster with peptide sequences that start with 2 TQT. Okay. And here I have other 2 subsets of clusters that were assigned to the same sequence. But actually, I can see here that the distances are much, the similarities are much smaller, right? So here I have 3 peptides that have been identified or matched to the same peptide sequence, but actually they seem to be quite different. And we can have a quick look at this. And actually, maybe now is a good idea to have a look at this function here. Yeah. So the spectra of this package has another function called browse spectra. And so if I call browse spectra, and I have a window somewhere, where is it? Sorry, where did it show up? It's here. It's here. So this allows me to kind of browse, navigate through my full experiment. So this is my first spectrum out of the 13. You see here, 1 out of 13. And that spectrum, here's the retention time, the precursor M over Z, was assigned this sequence. And here I have this MS2 spectrum with the matched fragment ions. And so here I navigate to the second one, the third one. And now that I have a kind of a small subset of peptides of interest, I can easily, that doesn't make sense for 80 or 90,000 scans. It would be possible, but it would not be very useful. But here now I can kind of navigate from one to another. And that's also something that is quite interesting once you have reduced your raw data to a subset of scans, spectra that you want to investigate in greater detail. And that's with the browse spectra function from the SpectraFix package. But let me regenerate my heat map here. Where is it? Sorry, too many things open. Yes, of course, I first need to kill this. Here it is. So let's quickly have a look. We have a couple of minutes left. What happens to here inside this subcluster of peptides that were matched to the same sequence, but we see that we have kind of these two that are grouped together, and there's these three others that were grouped together. But they all have been matched to the same sequence. So what I'm going to do is that among I'm going to I'm going to focus on those that have a sequence that starts with DLP. Right? These are those that I'm interested in. You know, the three first amino acids are DLP, and these are scans 2, 4, 9, 10, 13. And let's look at the information of these three using the same type of code that we had before, spectral data, now of my subset, and only the elements 2, 9, 2, 4, 9, 10, 13. And I'm going to look at the precursor charge. So all these precursors had a charge of 2, the precursor M over Z value. And here now I suddenly see that I have two different M over Z values for these precursors. And if I look at the modification name, I see that 3 out of these had modifications and 2 did not. So these 3 here, those that had modifications, and the 2 at the top here that form this other cluster are those that did not have any modification. And if I want to compare these in detail, I can also produce spectra, mirror visualization of these spectra, where I have one spectrum. So this one was the 4th one, and then the 9th one. Okay, to really kind of compare these spectra 2 by 2. Okay. So it was probably quite quick at the end, but what I wanted to demonstrate is that even though we can load this large data set, and here I only use 3 files, and this will easily scale to tens or even hundreds of files, again, because we use clever tricks to avoid overloading the memory of our data set. You know, what would take the most time would be to download these 100 files and not to load these 100 files in R. But sometimes we can focus on some scans of interest. Here I chose the 13 scans from a protein, and then I can cluster these MS scans based on distances calculated using the fragment M over Z values. And we kind of focus on one subset of these and identify that we had 2 subclusters with and without modifications. But these are the kind of things that you could do, or kind of selecting any scans of interest and not necessarily only those that are identified. So can you also load top-down data and use these packages? As long as you can export your data to ML files, then yes, you will also be able to manipulate these data. You will also need to have identification data. And again, if you can export these to NZID files, then you should be able to do everything I described with any type of data. The only requirement is that the data need to be exported in one of these open formats to be able to handle these. And so also a really good question here. If the purpose of this tool is to record how MS data is analyzed so that it is reproducible, at what stage should we implement it? Right after searching and identifying proteins or before additional analysis? Also, how can we incorporate other tools like MS-Stats TMT into the script? And so this really depends, what are you aiming for? So here I focused on raw data, but you could proceed with that analysis, with that concept of an experiment, and also add quantitative data that you would use for your statistical analysis, for example, using MS-Stats TMT if you have TMT data. So really that very much depends on, where do you want to invest your time to make sure that you understand and are in a position to reproduce all the steps of your analysis. Sometimes, like I showed here, this could be at the very beginning when you have the raw data. But in other cases, you might not have access to the raw data, or you might want to focus on the quantitative data and on the statistical analysis, for example, using MS-Stats TMT. And so in that case, you could start later on. I think that will very much depend on what your priorities are. There are cases where I start from the beginning, as I showed here, and there are cases where, for example, I get a spreadsheet with quantitative data that was produced by, I don't know, MaxQuant, Protein Discover, any piece of software. And then I would kind of focus on the reproducible aspects later on. This really depends on your priorities, what is important for you, and also, of course, on the time you have. Can you invest time to go back to the raw data, or do you prefer to invest the time and your skills on the analysis of the quantitative data? Voila. So I see it's 6:30, so I think I have used all the time that I was allowed to use. And so I will add all the code that I wrote here to the tool page so that you have access to it. But kind of none of this code is new. All that code is available. Where is it available? Let me check. Is available here for mass spectrometry docs. Yeah. Is available in this tutorial with lots of details, such as introduction about kind of how does mass spectrometry work. But I'm sure all of you know this. Accessing data. How can we access data? And here's an example of using the RPX package. There is a chapter on raw data, right? Raw mass spectrometry data, and you will recognize the figure that I used before. Description of the spectral class. Much more about visualization, right? How to reproduce this figure, where we kind of delve into one MS1 spectrum in detail, and then look at all the MS2 spectra that were produced by selecting 10 precursor ions here. There is a section about identification data, quantitation data, and so on. And these tutorials are being updated on a regular basis. Okay, so I will look at errors that were posted in the chat and in the Q&A, and kind of try to address this directly in the chat. Otherwise, I hope this was useful, and thank you very much for your interest.
Thank you very much, Laurent, for the presentation. Demoing things live like that is never easy, and Laurent is really an expert in that. Thank you so much. So we now have time for a 10-minute break. please take this time to take a break, but also to ask Laurent additional questions in the Q&A. So Laurent kindly agreed to stay for a little bit of time. Well, it's getting late in Europe, but Laurent will stay a little bit during the break to answer questions that you can have in the Q&A box. Other than that, we will be back in 10 minutes. So 12:45 Eastern, quarter before the hour, for the next presentation by Nuno Bandiera and Meena Choi. So please don't go away too far and come back at 12:45. Thank you very much.
Laurent, can I ask you a question?
So, yes, go ahead.
So this is it's a curiosity. I work a lot with mobility mass spectrometry data. Are there already any packages that can help me visualizing, let's say, specifically it is MSMS spectra where the fragments have been separated by ion mobility? Because I would like to compare multiple spectra to check if there might be some ion mobility differences for MULI5 fragments.
So among these kind of variables, these annotations that you have for spectra, you would also have an ion mobility time among these. So you could also do filtering based on ion mobility. So yes, that's not the kind of data that I use on a regular basis, but there are people that analyze ion mobility data using these packages. So yes, it should be possible.
Okay. All right.
And if you have more specific questions, or if you have a data set and you want to try, feel free to post questions on the Spectra page, on the Spectra GitHub page, open issue, and kind of ask more specific questions, and we're always happy to help out.
Okay, thank you very much.
You're welcome. Okay. Answer. So here.
Laurent, I can try giving you feedback on the issues that I'm having for installing. Because I saw that, I tried removing the package and installing the package of SAS. And still, I try to do the run of the installation of all of the packages, and it gets blocked at some point. I guess it doesn't finish. And whenever I try to load any of the RPX or following dependencies, it's failing. So I was not able to follow. But I'm not sure what that SAS thing is. And when I installed it, it says that it's, well, doing the most recent one in there, 0.4, but it's still trying to get the 0.31. I'm not sure what's going on.
Can you hear me now?
Yes, I can hear you now.
I'm not sure if we are live or not, but so
yes,
I was
you
saying,
are.
Okay, I'm not sure about this SAS dependency. I'm not sure it's one of mine. So maybe if you restart your R session and try again. Yeah.
Okay.
Otherwise, we can also take it offline, and I can have a look and try to help, with that specific issue if you share your screen.
Okay. Thank you.
Okay, it is time for us to come back to the second part of our discussion. And we continue on the topic of reproducible research, except that now for the second part, instead of focusing specifically on R, we will talk about reproducible research more generally when we have workflows that use tools which potentially rely on different programming languages, different types of experimental workflows, and different data analysis strategies. So it is my great pleasure to welcome Nuno Bandiera and also Meena Choi, who will do a team of two presenters presentation. So let me first welcome Nuno. So Nuno has a PhD in computer science and engineering from University of California, San Diego. So after finishing his PhD, he stayed in the sunny San Diego, and he is now associate professor with a joint appointment in computer science and engineering, and also in the Skaggs School of Pharmacy and Pharmaceutical Sciences. And Nuno is executive director of the Center for Computational Mass Spectrometry, and he is the principal investigator in the effort Massive, which facilitates data sharing and community-wide interpretation of publicly available mass spectrometry data. So Nuno has been recognized extensively for his efforts. In particular, this year, Nuno received the Gilbert Ahmed Computational Proteomics Award of the US Human Proteome Organization. And just very recently, he is this year the recipient of the Beeman Medal of the American Society for Mass Spectrometry. And we are very fortunate to have a chance to hear from Nuno today. And welcome, Nuno. Thank you very much for being part of May Institute.
Thanks, Olga, for the very nice introduction and for the invitation to be here. This is really a prime forum for talking about mass spectrometry and talking about the sorts of things that we do. And it really is my pleasure to be a part of this program and to be able to talk to all of you here today about the sorts of things we're doing with regards to, you know, helping enable reproducibility, help promote reproducible analysis of data. And in connection to this topic today, now talking about it from the perspective of the data and how do we make the data available and how do we help share the analyses that were done at the time so that you can then, you know, learn as much as you can, not just about the results that are presented, but also about the way in which the analyses were done. So in the context of this topic, I'll start by giving a bit of an idea of where we're coming from with Massive. And then I'll talk about the Massive Quant effort that we put together to go more in this direction of having reproducible research, not just at the ID level, but also at the level of quant, which obviously is critical for the analysis of biological data. So you might have seen this from the outside. Know that there is a proteome exchange consortium that is now pretty well known in terms of data deposition, that when papers are published, data has to go somewhere that is independent of the researchers that derive those data sets. And this was started about 10 years ago. And you can see that this slide, which I keep at this point for historical reasons, in terms of how things were in 2016, 5 years ago, that we can see then in contrast to that, what does it look like today? So 5 years ago, we had 4,500 public data sets, and it was a quest to get it to that point when we started. Things moved very slowly. But since then, as you can see, things have changed very quickly. So in 2019, when we presented Massive Quant effort for the first time, things were close to about twice as many data sets as originally deposited. And today, in the last 2 years, it's already almost doubled again to, you know, almost 3 times, almost 4 times as many data sets as 5 years ago. So things are growing really quickly and data is becoming available really quickly. So the question we want to consider is, how do we make the most of this data? How do we make it possible to release this data in a way that can be most informative to support future research as well as to explain the research that was originally associated with those data sets. So in general, the view that a fair number of people have of how repositories work is that you do your work, and at the end, right before submitting your paper, the journal has that nagging guideline, and you just have to put it somewhere in a repository, and it just goes up to stay there. And then, maybe a couple of people later down the road might be able to download that. But at Massive, we wanted to challenge this view, because as it is today, because of the way that data is deposited, most of the time you actually have a pretty challenging task if we want to reuse anything of the data that was deposited, because data sets are deposited as raw data, and the analysis results, if any, are deposited in a proprietary format. And that usually makes it complicated for others to open and to be able to see what was done in those analyses. So, the picture, unfortunately, looks sort of like this in general, that only, 1 in 20 data sets roughly has reusable identifications, in the sense that they're released in an open format that you can open with tools, open source tools. And most of them essentially have no reusable quant data. If deposited, it's in a format that you can't easily look at. So Massive from the beginning was designed to restructure the data and to help it be reanalyzed, to be converted to open formats whenever we can. We try to convert the formats that are submitted by the users into open standard formats that you can then open in other tools and use standard ways to analyze those data. The data can be automatically reanalyzed, and we have done quite a sure amount of reanalysis, and we'll talk a little bit about it today. And then we want to provide advanced visualization interfaces that you can use to analyze the data without having to download it. You're very welcome to download it if you'd like, but since we're talking about hundreds of terabytes of data, it is preferable if there are ways to help you interact with those large volumes of data without having to download it, because it's just too much to download. With the goal being that hopefully this can help integrate this data into your research process, rather than being something that comes only after the whole research is finished and you're just needing to get an identifier to submit your publication to a journal. So there are a few things that you'll see when you come to the Massive homepage, and the most common two things are shown right at the top of that page. And that is, if you want to browse the data sets that are available and search them by metadata, that's one of the options that's available at the top. And the other one is to submit your data. Those are the most common operations that we see. But I wanted to also mention what it is like to have a data set when it's submitted to Massive. And there are a couple of things that you see on a data set page when you get to it, because the most important point here is that a data set is not static. It is one thing when it is first submitted, but it can and does evolve over time. So the first thing is you get the identifiers for a data set. And in this case, we see that this is a data set that was actually originally released in the NCI, or National Cancer Institute, Proteomic Data Commons. And then it was imported into the Proteome Exchange Consortium with this Massive identifier, and thus assign that Proteome Exchange identifier. So you can find data sets that are present in multiple repositories. Definitely, even if data was originally deposited to PRIDE, or in this case, to the Proteomics Data Commons, it can still be imported into Massive for reanalysis and reintegration into these tools. And in particular, for reanalysis with Massive Quant, which is the topic that we'll get to here today. So we do provide the standard operations that you expect to see on data sets. You can add or update metadata on your data set. You can add publications to a data set. We also provide the ability to add files to data sets. Not all repositories let you do that, but if you need to replace a file in your original data set, you're able to do that. You cannot delete things that are in data sets. Data sets are immutable. So once published, then they can't be actually once submitted, not just once published, because during the review process, it also should not change. So once a data set is created, you can't delete anything from it, but you can add things to it. But we also allow the other kinds of options that allow you to expand on the things that you can do with the data set. So in particular, you can convert the spectra to open formats, which is an important step that is necessary for you to reanalyze it with open source software. So this can be done online for you. You don't have to download and do that. And once the spectra are converted, then you can reanalyze it with a variety of different tools. There are, your standard database search tools, library search tools, proteogenomic search tools, open modification search tools, so on and so forth. You can upload your own. If you did an offline analysis, you can also upload your own and attach it to a data set. And we'll hear more today about how to do this for quantitative results. But you can also run MaxQuant and MSStats directly on the system, which is something that has been a great interaction we've had with Olga's group to put together this ability to do the quantitative analysis of the mass spec data directly online without having to download the data and figure out just how to do everything offline. You can actually do these analyses online now also.
No, there's a question. Can we change the name of submitted files?
I'm sorry. Where was the question?
I
had the answered open, not the open ones. I'm sorry. Thank you for reminding me of that. Can we change the names of submitted files? Interesting question. Actually, interesting as it sounds, I don't think it's ever come up. Generally speaking, we wouldn't, but I can imagine cases where it does make sense. So it doesn't actually change the file. So I would venture to say yes. I think that's an edge case that we hadn't considered. But I know that in some cases, the metadata files have some file names that differ from the file names on the data set. And definitely corrections like that would be welcome. So I would go ahead and say yes, because that does not change the raw data. And that is really the intent of this guideline for the whole Proteome Exchange Consortium, is that the data itself does not change. But if you're just changing the file names, presumably to make it clearer what the data set submission is, then I would think that makes sense to allow to do that. It would have to be something that we would have to do. It would have to be an interaction that happens over email with us. So the system doesn't provide that option online, but we would find a way to do it, especially if it is to clarify the purpose and the content of the data set. Absolutely, I think it would make sense. Thank you for the question. So I was going from the ways in which we can reanalyze, and then these reanalyses are added on the system. And in this case, for this particular data set we're looking at, there's already a series of reanalyses of different kinds of tools and with different kinds of results, and they all appear attached to the data set that as was originally submitted. So you don't have to go to a different place. As long as you can see the page for the data set, you'll see all the reanalyses that have happened for the data set. And then what can you do with it? So there are tools provided in the system that allow you to compare reanalyses. And that helps because you may want to compare different tools and you want to see how the gains and the sensitivity of the different tools compare. You can compare different protocols. You can compare different samples to see if you're finding the same proteins or not the same proteins in different data sets. And the way that this works, you can select results from that are attached to data sets, or you can select any of your private searches to compare to those data set results. And then you get results at the spectrum, peptide, and protein levels, where you can see how the two sets of results compare. Now, if you want to go beyond the pairwise analysis and comparison of results, you can actually search all the identifications that are submitted in Massive. And as it is today, it's originally it was over 300 million, and technically it's still over 300 million, but now it's over 500 million identifications that are available in a system from dozens of terabytes of data. And you can use those to look for any protein that you're interested in, or any peptide that you're interested in. You can find all the data sets in which it occurs, and you can find all the modification versions in which a peptide occurs, and so on and so forth. So it gives you the ability to search across all of the data in the system. And if you want to use a distilled version of all these searches to help with new searches, then we have the Massive KB, which stands for Massive Knowledge Base Spectral Library, that you can download with over 2 million precursors of reference spectra. And there's a Protein Explorer interface that allows you to see all of this information in a protein-centric manner, instead of just being more data-centric, where you're looking at data sets and you're looking at the data sets metadata, or the spectrum-centric view of the spectral libraries. Here it's a protein-centric access into the system, where you come with your protein of interest and you can see all the information that's available for that protein. So it is in this context that we come to Massive Quant, in the understanding that quantification obviously is one of the most important, if not the most important, kind of analysis that you can make on proteomics data sets. And we really needed to have a way to represent the complexity of the protocols that are used for the analysis of this data. And as you can see, just briefly illustrated here on this slide, there's very many different ways in which you can combine tools for identification and tools for quantification to then do the statistical analysis to get the results, the quantitative results and the differential abundance results that you then use to understand the biology in the samples that the data sets come from. So there were many different ways in which these protocols happen, and we see here different studies that combined these different ways of analyzing the data in different ways. And each path that you see from the top to the bottom are different ways in which the data sets at the top were analyzed to result in different reanalyses. So unfortunately, we don't even have standard formats for all of these. There are some standard formats for ID, but for quantification, that is actually something that is even still being discussed, as well as for the metadata that would be necessary to describe the samples and to give you a chance to run the quantitative analysis. You need to know which files go with which samples and which conditions so that you can then do the analysis. So the approach that we took for Massive Quant was to try to come up with a way to capture all of this information so that we can begin to share these studies in a meaningful way that has enough information to understand how the studies were actually conducted. So with this goal in mind, we teamed up with Olga and Meena and put together the Massive Quant resource, which builds on Massive's ability to store data sets and to represent data sets, and adds the additional things that we need to represent quantitative data sets, where we aim to capture the metadata that's necessary for these analyses, as well as the scripts and the results that are derived along the way, as well as the final results, of course, of the quantitative analysis. So as it is today, we are at slightly over 200 reanalyses that are quantitative and give you differential abundance results of 64 data sets across a variety of different to interacting with all of you to see how we can facilitate the sharing of your analyses and how we can make these results more useful for you. So it's great to be here today and have the opportunity to have that conversation. So there's a question here of whether this is limited to DDA, and the answer is no, it is not. The massive quant resource can take results from any kind of quantitative proteomics experiment. In particular, it can also take DIA results or targeted analysis results. As long as the quantitative tables are available, then we can have that be the quantitative information that's made available for the data set or for the reanalysis, and include that as the result, as the quantitative table. Now, we wouldn't have tools to process that kind of data. So there are some tools in Massive to process DIA data, but note that you don't have to use any of our tools. You could have done all of your analysis offline and then just submit the files that you got from your offline analysis. If you wish to use some of the tools that are available online to assist with the analysis, then at this point, they are more DDA centric. So for the both label-free and isobaric labeling experiments in DDA mode, we can actually support the full analysis being done online, either on public data or on your private data. But for the other kinds of data types, such as DIA and targeted, Massive Quant still supports it as a submission, but we don't have the tools to do it online. But it's still supported and definitely welcome, because at the end of the day, it is the differential abundance results and the proteomics results that are most important, and we'd definitely be glad to have them. So some of these reanalyses here already are of DIA data sets and of targeted data. So we could definitely highlight those as examples that could be used to start new submissions if you're interested in going in that direction. So the sets of new features that had to be developed for this system essentially fell into three major categories. The first one being we needed to have a new data set format. There's another question here. Let's see. Staggered DI data, okay for massive. Is it better to submit non-demultiplexed raw or converted plus demultiplexed MZML? Great question. So I would actually recommend submit both. That you can submit both the raw files as well as the deconvoluted and converted, demultiplexed in this case, MZMLs. Both would be welcome. And in some cases, we have DIA data analyzed with DIA umpire. And that's an example where we have both. We would have both the original swath files as well as the deconvoluted MS2 files. And there's actually more than one. It outputs more than one. All of those can be submitted in separate collections. So when a data set is submitted, there are different subsets of the data. There's a collection for raw data, and then there's a collection for converted files in open formats or standard formats. And that's where the converted and demultiplexed data would go, with the raw data going in the raw collection. But both would be the best, just to make sure that we have the best record of what was originally done. Because as you know, there's different ways to convert the data. Sometimes there's centroiding, sometimes there isn't. And there's definitely different ways to demultiplex DIA data. So the best way to make sure we have a record of what was done would be to have both of them in there. The other question is it possible to retrieve a large set of proteins peptides to generate a spectral library with massive tools? If so, are there limitations? So let me see, two different ways to answer this. The first one being, if we're talking about human, we already have a spectral library that covers a lot of the data that is available out there. And it has actually, I mentioned earlier in the talk that it was 2 million precursors. It has actually been expanded recently, and it's now almost at 4 million precursors. So for human, we have a pretty comprehensive spectral library already in place. Now, if we're not talking about human, or we're talking about a situation where you want a smaller spectral library for a specific kind of tissue or for a specific kind of sample, then there are workflows in the system that allow you to generate those spectral libraries from any set of data sets for which we have reanalysis results. And if we don't have the reanalysis results, then you can, then those can be obtained. Then there are workflows to get those reanalysis results. So it is possible with, it is possible in both cases. So if it is using the whole human library that's already available to download, and it's a simple step to just essentially download it. And if you want to make new libraries for either different species or for different specific conditions, then it is also possible to do that, because the exact same workflows that we use to build our libraries are publicly available for anyone to make other libraries. And essentially with the difference being what the input data is for those other libraries. Are there limitations? I'm sure there are. I don't want to say that there's low limitations and anything is possible, but I can tell you that in terms of volume, our human libraries, start with dozens of terabytes of data, and they start from, hundreds of millions of PSMs. So the system is able to scale to those levels. I guess what I would say in terms of limitations at this point is you would need to start from DDA data. And right now it is expecting either CID or HCD type of spectra. There are some filters that have to do with explain intensity. So if you're using ETD or ETHCD or different kinds of fragmentation modes, the workflows currently are not equipped to deal with that. So I would say, as long as you stay within DDA and you stay within CID, HCD, high res or low res is fine, then you should be able to build those. So great questions, everyone. Let me then go back here and say that in terms of the dataset formats, what we needed to add to the infrastructure we already had to support raw data and open data and reanalysis, identifications reanalysis, we wanted to be able to support the kind of data that's important for the quantitative analysis. So we wanted to be able to capture the metadata for those data sets. And because, for reason one, there was no standard format to represent that. And reason two, we want to make this an easy next step for people who are already doing the quantitative analysis. We actually made this metadata format be modeled after the MSStats input format. So the same file that you learned how to configure here, or you already knew from before, it contains all the critical information necessary to describe how the data, the files that you have in the data set or in your study, relate to the samples and replicates in your experimental design. So to make things easier, we just essentially adopted that format and said, if you can submit, if you can submit that metadata, then you can make a quantitative data set or reanalysis submission. The other two collections contain the quantification results. These would be either the peptide abundances or protein abundances that you get from your quantitative analysis. Could also be TMT ratios or other isobaric labeling ratios. And then there's a final collection for statistics that contains the statistical analysis of the quantitative data. And that is where you would see whether things are differentially abundant or not. And if they are, at what level of significance. That's essentially the distinction between these two. One of them is the quantification results. The other one is fold variations and statistical significance of those fold variations. And then finally, as I already alluded a little bit earlier, we also wanted to provide executable workflows that you can use directly in the system to run the analysis, to facilitate running your own analysis, as well as the analysis of public data. So these are workflows that you can use for both private and public data. So if you upload your data to Massive, you can actually work with it and analyze it in the process of your research project, and none of it is public. You actually have to explicitly go through a series of steps to make anything public. So you'll never make anything public by mistake. And it is ideally the better way to go and do things if the tools are available in the system. We'd be glad to support that research process directly online. And one of the reasons I really like to do it online, one is that it makes it easier for me, that I don't have to write things on a command line. And if I'm doing it for many reanalyses, it's easy to just clone a job and rerun the analysis. But one other reason is that these online tools also provide you with interactive results views that show you, in this particular case, the results of the MS stats analysis. You can see all the differential abundance results. And if you click on the little graphic icon for each row, you get to see how the different abundances split against the different conditions, and you get to see the box plots for the different comparisons that were done for the particular analyte in that row, which in this case is the protein level analytes. So one last point I wanted to make here is that a recent batch of reanalysis that has been included in Massive Quant derived from efforts in this past year for the analysis of SARS-CoV-2 and COVID-19 data sets, as well as data sets of related viruses. And these cover a variety of different experimental types, as well as different types of omics. There's also metabolomics and lipidomics data included in this Corona MassKB resource, reanalysis including PTMs and ID, but most importantly in the context here today, also quantitative reanalysis, including MS stats results that are now available and can be accessed through either system. You can access it through the Corona MassKB resource, or you can access it through the Massive Quant resource. So with that, I'll just stop here and give us a moment to ponder this high-level view of what the system does. And then Meena can tell us a little bit more about how specifically you can submit reanalysis into the system and interact with the system for either submission of data sets or reanalysis. If we use the executable workflows online, can the results be shared with collaborators online? Absolutely. It's actually very simple to do that. When you get your results, there is a link that has a random string in it. And if you just copy paste that link and email it to anyone, they can see the results that you derived. And you don't have to remember that link, because in your account, we keep track of all the jobs that you ran. And if you click on that list, you can see all your past jobs. So you don't have to go and memorize or save it. But yes, it's very easy to share results with anyone online. In the online analysis of the workflows are somehow optimized? You mean automated or authorized? Or as users?
Automatic.
Automatic.
Yeah.
Yes. So there are definitely, as users can work with the tools offline like we do, online like we do offline, choosing all the different parameters. Yes. So, okay. Yes. Maybe. Depends on the parameters. For most tools, there is an input form in which you can select values from many parameters. But for a tool that is as sophisticated as MS Stats, there's always going to be a bit of a trade-off. So if your study is not a standard type of study, if it's a less common kind of study, then there may be things that could be done offline that can't be done online. But for most studies, in particular case control types of studies, multiple group types of studies, those sorts of things you can definitely configure and run online. And you can select things, you can select filters like minimum number of peptides per protein, minimum number of bioreplicates per group. There are things like that that you can configure in the input forms for the tools. So the goal is to be able to capture maybe 90% of all the analysis that you can do with those tools. But we do recognize that the 100% is not possible, because in some cases, you need some specialized types of pre-processing of the data that currently may not be supported. But in general, I would imagine that the majority of cases would be supported. Now, before transitioning, I just wanted to mention something very quickly that unfortunately I need to teach at the same time as this session. But it pains me because I really like the fact that we are here interacting with you and we can hear your questions. So please, if there's anything that I'm not able to answer here and you'd like to consider, or is something that is in your mind before deciding whether to move forward and try the system, please do make sure to contact me or contact us if needed to keep the conversation going. So quickly, there's another question here. Is it possible to implement massive tools locally? So the workflows are made available as open source. However, there's a number of things that are done at Massive that are based on accessing public data. So from that perspective, it's kind of intertwined with the system. But when it comes to the algorithms, we definitely make an effort to make those workflows available to run locally if you'd prefer to do it that way. Or to look at the source code, absolutely. We do make an effort to release them all as open source.
Nuno, thank you very much for the presentation, and thank you for joining us. We know your teaching schedule was changed at the last minute, so that's what happened. We are very grateful that you could make it regardless of that. And indeed, for everyone, please don't hesitate to reach out to Nuno with more detailed questions after this session. But at this point, let me welcome back Meena. So as you have heard from the previous session, so Meena is currently a research scientist at Genentech, working on all things quantitative mass spectrometry. And before joining Genentech, Meena was the lead developer of MS Stats. But in addition to that, Meena also was the leader of this effort that integrated the workflows in MS Stats with the data and workflows in Massive Quant. And so for the rest of the time today, we will talk about some specific details of how we can use quantitative workflows within Massive Quant. Thank you very much, Meena.
Thank you, Olga. So we had a really great session this morning, talk about the reproducibility with the Laurent, and then the whole pictures of the massive and massive quanta from Nuno. So the goal for this section is I want to introduce the massive and the massive quant a little bit more detail. So all the aspect of what the Nuno mentioned, I will just show the briefly, and then give you all the sources you can follow later. And then I will not use the present mode because I just want to go back and forth. But if you have a difficulty to read my screen, please let me know. Okay, so massive look like this. So let's go to the massive first. So I think that you can get the slide, and then this is the link. You can go to the massive. This is the first page. So here's the massive one. If we scroll down, you can see there's many things that we can do it. We can access the public data set. We can submit the data. We can search identification, and so on. And then the bottom, we can see here's the message quant. So here, if you click the link, message quant, this is how the message quant web page. At the beginning, there's a summary of statistics of the data set. So for example, here we have a 64 public data set, which include the metadata and then quantification data. And then there are more than 200 quant reanalysis data set is there. And then among that data set, the number of the data set by species, so there are 36 data set is for experiment with the human, and then others is the mice and something else. And then if you scroll down, there's a list of the item what we can do in the message quant. So we can browse the public data set. We can submit the reanalysis. We can access the reanalysis. And then we can use the MSF workflow in the message quant. And then we can compare the workflow. We can compare the MSF output in the message quant and so on. So each section you can click. So for example, if you want to browse the public data set, you can click this, or you can click the documentation. So there's each step, each task, we have a documentation link. And also we have a video tutorial. So if you wanna the summary analysis, you can check the documentation. Let's click this, how it look like. There's the documentation. You can just follow one by one. So here, so upload the data, and then click next. You have to add the analysis, and then something else. So there's tons of the documentation there. Also, if you click the video tutorials, we have a video. I will just stop doing playing. But in the Olga Vitex lab, there are, so for example, this one is for 15. So I will just record my screenshot to how to, you can submit the reanalysis. So you can just follow each step over there, but I will go over shortly today. So first step. So for message quant, what we can do, we can just browse the public data and submit the analysis. The browse of public data is quite important for like the computational person like me, because sometimes we want to play around the data set from publication. So they submit the data somewhere, but we want to access this one, right? So, or we want to search in. I want a human data set for specific acquisition type, and then actually we can search this. So this is the goal for the browse the public data. Also, some analysis means we don't need a road, we don't need a new experiment. We just want to use the public data experiment, but we want to reanalyze that data, and then we want to submit it. So to share it, like just for multiple purpose, maybe that is for maybe also another computational of the paper to propose, or if we want to share this one, or we want to compare in the different other result in the message quant. Also, we can access the reanalysis, which means we can access the reanalysis from others. And then we can also just look at how they're doing and etc. Also, we can do the MS step workflow in the message quant and then compare it. And then there's a lot of the documentation there. Then let's see how we can browse the public data set. So here's the message quant. And then let's click the browser data set. So this is how it look like. So message quant data, there's a big tables to show, to list up the whole data set which require, which is satisfy the requirement for the message quant here. So you can see whole the list of the tables, right? And how we can make your data set available in the message quant is if you have the quantification data result, means quantification data, and then you have a study design, means another metadata, what I'm gonna talk to you soon. And then the result of differential abundance result. If you submit 3 data set, it will gonna automatically show up in the message quant. So for example, here, total 64 data set already submit this data. And then the message quant recognize this data set have this information. And then here it is. So for example, here, this data, I have a keyword. This one is the DI data. And then for Reproducible, for the specific the research question. And then they have this one. And then from metadata, they automatically count this experiment have 17 different conditions. And then they have 42 biological replicate there. And then each one they have up to 5 technical replicate. And then they have 279 proteins are quantified, and then among them 231 is a differential abundance of proteins. So, and then you can also see who is the PI, and then what kind of instrument we use, etc. And then here's another column called the reanalysis container. So I'm going to talk about what is this means, but which means in this data set, there are several reanalysis there or not. So for example, first one, they don't have it, but second one, they have another space to store the reanalysis. Sometimes they have a plus sign, means they have a multiple container to save more number of the reanalysis there and so on. So this is how it looked like. I also want to show this one. So this table is a filter by any data set with the quantification result and then differential abundance, real analysis, and the study design. There are also, we have a review level. So the bronze, silver, gold, the platinum, and so on. So let's click the platinum. So there are 25 platinum one, which means they have all the information, and then we confirm we actually can reproduce the result. So which means they have a fully documented data, and then we need all the requirement information to reproduce their result. So this is how you also can use this one. So then maybe I want to look at the select data set what we play around last couple of weeks. So yeah, let's see where we have this data, or maybe they have a next page. For example, here TMT10 control mixture is the data set the team play around yesterday. So you can also go there and then see all the output from there. Here we go. So this data set is what we play around. So I will click this one. So here, tidal SWAS data set from East over the time in the response to osmotic stress. So here's the description there, right? And then here, they also have a summary of all the data set in this the data set. And then you can see also we have a publication. They have an owner, which means the first the submitter of for the raw files. Raw files, the owner did not provide experiment design one or quantification data. However, later we submit the 22 reanalysis. So we can just also double check this one. And then bottom, you can see also the list of the whole reanalysis analysis. So we're gonna look at this one a little bit and then just also play around with what we can do it. So cell-based analysis, we can say the two things. One is submit the new data, new experiment, and then we can submit the reanalysis. The new data means we are going to submit the raw data, start from the raw data, and then we are going to submit the intermediate. You can submit the intermediate one. Submit the real analysis means raw files are somewhere there. So data set have their own unique massive ID. So this ID is assigned for each experiment, each data set. And then this data set said this is the public, so you can get the public data set. And also we have the proteome exchange, the ID as well. So if you already submit your data set in the proteome exchange, we actually, the massive also can import to your data if we have this ID, and then we can also, you can have also another ID for massive. So if you already have this MSD ID include the raw file, and then I just want to reanalysis, and then we want to submit it to, if you just go back to the holder step for data analysis, this is a kind of the brief step we can do it, right? Though we can do the sample preparation, and then we got the data, which I will call the primitive raw files. And then we have to do the spectral identification and then quantification. And then we can do the statistical analysis. Last couple of days, we talk about how we can do this statistical analysis step using the quantification data, right? And before, we also talk about how we can do identification and quantification using Skyline. This is what also we talk about last week and last week, right? Here is some tools or the file, like intermediate file input or output for each step. So for example, sample preparation, we actually don't have it, but after we got go through the sample to mass spec instrument, first we're gonna have a raw file, right? And then do the identification, we might need the SQL database, right? So this is also should be input for identification or quantification step, right? So raw file or FASTA file or something else will be just input for this identification step. So here's one of the example. If we use the max quant, so we just put this information in there, and then max quant output quite a lot of the intermediate file, right? Among them, the MQPAR XML or parameter txt is the parameter file, which record what kind of parameter we used in the version numbers and so on for the max quant. And then also one of the output we are going to use for statistical analysis, the evidence file or group protein group file. So we call this is the quantification file, right? To use the MSTER or MSTER TMT, we need this the quantification file. And then we probably use the R script if we use the R in locals. And then annotation, the CSV, we call this is another way the metadata, which means metadata include your experiment design. So up to here, it's not a big deal to each raw file from which human sample or their age or their group, right? But statistical analysis step, it is important. This, each row corresponding to what kind of sample we have to use this one. Example of the annotation. This is also what we practiced with the team yesterday. This information is the required. So for example, here, each run means each MS run. Each sample is corresponding to what kind of condition and then from which biological replicate, right? For TMT cases, it will be, we have probably the one more column called channel, right? Because each channel have a different sample information. So this is the we call the annotation, and then based on this, we count how many conditions we have, and then how many biological replicate we have, and how many technical replicate we have, and then also we also count how many runs we have, and etc. So to get the final the statistical analysis, we to get the same, we if we want to reproduce the same the result, actually we need the whole intermediate file, right? If you're missing one of them, actually we cannot get this one. So for example, if we missing one of the quantification data, definitely we cannot get this one, right? And also parameter file is also very important. If we have a different version of the other tools, maybe something is changes, we cannot get the same result, right? If you have a different parameter setting, definitely we are going to have a different output. So to get to full the reproducibility for this our final output, all this information is needed, and then we can save all the intermediate file into the massive quant. Then this is the for one analysis, one real analysis. But each step we can use the several different things, right? So for example, we have a one experiment, and then we have a one set of the raw file, but maybe we can use the different the searching and identification algorithm. And after that, we also might use the different processing tool to get the quantification. And then for statistical analysis part, we can use the MSStat. However, we can use also different the statistical method. For example, we talk about the bioconductor today, right? So if we go to the bioconductor, this one is to hold the storage to all different type of the R of the package, to give you the idea how you can find some other the R package available there or something. Here's a use, and then if you click the software, this one lists up the whole the package available in the bioconductor. So for example, if you click the software, if you click the research field, I will say research field, there's a proteomics. So there are 145 is R package related to proteomics one. And then this one is linked by number of the download and so on. So for example, Lima, also we can use the Lima for proteomics one. And then Laurent's the package. Yeah, Laurent, we can see the Laurent's the package quite often here, right? So you can see whole the R package, and there are many different way to you can analyze it, right? So each step, each based on each your decision for each step, output will be different. So, but we want to save this one more systematic way in the one place and easy to access it. We have a question. So in case we are using another processing tool, so where Skyline Spectral Mine, should we provide the software output in the MSD friendly format, and or we run the no. So the goal is we don't want to lean on the very specific software. So that's why we want to make everything so generalize it, right? So this is the one of the case just to show you the demo. So if you use the max quant, this is the output, right? But if you use the Skyline, Skyline doesn't report, evidence files. Skyline doesn't report the protein groups. So Skyline report the MS2 for the format, right? That will be the quantification, and then we can use it. They probably also report some different parameter file. Then we can use a different one. Spectral Mind also they have a different type of the quantification, and then also sometimes, so for example, Skyline they have a Skyline specific format dot sky or Spectrum dot SNE or Proteome Discoverer.pd. That is also output from their tools, right? We can also save it as a quantification widget, and then we can just bring back. So this is the kind of the one of the idea, but at each intermediate file format or file type might be different. You don't need to run everything through the max quant, but this is required to submit the intermediate file to get the final output. So as much as you can. So for example, log file, parameter file, it is very important to record your reanalysis. Right. So this is the whole idea. So we want to save all your selection in each step. So how the massive quant structuralize it is a massive quant support the branch structures. So at the top, the high level, we are start from the data set. It could be one experiment or one publication. And then massive quant give the ID start MSV number 000 or something like this. So this is the ID for the specific the data set. Under the data set, we can make a several container, right? So under the data set, we can save a container, and then this container have also new ID start with the RMSV. So for example, I just want to analyze that specific data is already available in there. I will make the my container there, right? Or maybe Ting later, Ting also do the reanalysis later, and then she can make a container, her own container under the data set, right? So we have a two separate container. And each container, maybe I want to try the several different parameter setting to analyze the data set, right? Then each parameter setting, we have a different analysis. Then we have another unique ID for the reanalysis, which is RMSV dot number dot X. So we have a branch structured data set, and then we can make multiple container there. And then each the container we can make a reanalysis. And each reanalysis we have a one space kind of the folder, and then we can put all the intermediate file there. And then we have an ID and link. So whenever you want, you can just give this link to others, or we can just save this link for the future purpose. So here is the one of the example. So this is the one of the data set, and I just make a two container. And then for first container, I reanalyze a set of the raw files using different processing tools. So first of all, this reanalysis processed by Skyline. And then another reanalysis is processed by MaxQuant. And then another one we processed by the Progenesis, and then also with a different setting. So we can, from the one set of the raw file, there are 10 different reanalysis. I can save it up there. And then I also make another container, how I can use the MSDAT the parameter. The first one is the different processing tools and their parameter setting. Second container, I've made how I can use the different parameter setting for MSDAT. But how you can organize this is quite up to you. It's a kind of how you organize the folder. But the important thing is the message quant to have a support this branch structure. You can save all the output as much as you can. Also, I saw some other cases is data set is for one publication. But sometimes one publication, we have a several different set of the experiment, right? It's up to you. You can make each experiment, you can make a new massive ID, or to submit the paper or to share the data set, make a data set for publication level. And then in container, we can submit the each experiment. In that cases, we have a different set of the raw file for in our container, right? But the data set level, they're gonna contain the whole set of the raw file there. But in the container, we can select which raw file we use for this specific analysis and so on. So, okay, so this is how you can organize it. But to submit the reanalyze, how we can do it, and then what we needed. To submit the new experiment, there's the huge documentation there. I'm going to show that briefly. To add the reanalysis to massive quant in the existing data set, three things are required. One is the quantification data. For example, MS did input, which is a skyline output, what we practice, which means a feature level data. This is a starting point. I want to use this one for statistical analysis. Metadata is exactly what we show. It should include the information of your experiment design for each run from which samples. So this is the quite simple cases, but and then we use the east one. However, if you have a human data set and age, gender, some other really good important information we use, definitely you can add this information in the column. But this 3 column is the required. And then the last one is the output of differential abundance analysis. Definitely we need the log to 4 changes, and then p-value or just p-value column is required for this one. If you use some different statistical analysis, definitely you can submit the output in here, and then you can fully document it in the real analysis, the branch. Other thing is the parameter setting file is not requirement. However, I strongly recommend to submit this additional information. Also, the R script or log file to record how you use the R or Python and so on. Definitely, it would be good to submit it there. Okay, so you have all the set of the data set, and then I'm ready to submit it. And then we never used the message one before. What should we do is the 3 steps. First, you need to register the massive account. And then you have to put all the file in local computer to the massive space. Then you can do it. So here's the video and then documentation, but I will gonna show you how to do this. So when we go to the massive page, you can see here top right the user and the sign. So I already made my account, so I can just click sign in. But if you don't have it registered, this is the only information needed. So no extra step. Just put save your user name and the email, password, and submit, and that's it. So you can start to use it. So I will sign it, and then please use the login to use our workflow. The successful login, right? So now here we go. To submit the new data set, submit new data, your data. And then there's nothing here, but it will take a few seconds to us show whole things. Here, the workflow selection, the workflow is what the Nuno said today. Here is already selected the massive data set submission workflow. So we can just start from there if you want to submit the new data. However, if you click the button, here's the whole list of the workflow what the message want to support. So for example, here you can add the file, convert. You can use the MS-DEC workflow in there. MS-DEC comparison workflow, we can use it. Add comment, add your analysis, pride import, right? So if you know the pride ID, you can just import your data set into the massive. So this is the workflow and access, etc. Right? So to submit the new data set here, and then you need to give them. So here's a data set metadata, means experiment third part. So species, instrument, who is the PI, and then description, and so on. So as I mentioned, whole documentation is there. Let's see some or one of the document there. Right, so here's as what we showed there. So really detailed step there. You can just follow it, how you can select the species, and so on, right? One thing I want to point out the submission file category. So we have a several different types of files. Someone ask about the MGML, MDID, what kind of information we needed for ID part. So here's the list. So for example, require for complete submit, you need a peak list of file, and then this is the file type, and then return to files, and then requirement for all submission, which means this is really required. If we don't have a raw file, we cannot set up the submit a new experiment, right? And then also searching engine file, and then also the optional one. So here's the optional one, and so on. So I recommend to check this part if you want to submit a new one. And then, okay, so now we have a account for massive, and then let's go to the massive quant, right? And then I want to submit the reanalysis. To submit the reanalysis, we have to know the massive ID first, the ID for existing data set you submitted, and also we have to um, load the files what we got from local computer to the massive space. In that case, there are several ways to upload it. There's a documentation there, but I use the FileZilla, the FTP one. So here is the FileZilla. So here you have to type massive ucsd.edu, and then you have to use the massive quant ID you generated, and then password, and then connect. This is okay, right? Then we have a laptop panel and then right panel. The laptop panel is my local, my computer. The right panel is a space of your account in the massive. So we have to transfer file to the right side, right? If you are the first time to use the massive, probably it will be empty. But I put the many data set already there, so it looked like a lot. But it's the same as what you can play around. So for example, I want to submit a select data set what we practice last couple of days to the massive quant one. So here's a MySpace. So you can just organize as much as you can. Just recommend to name it that can help you to easy to find it. So for example, here you can right click and then create the directory, right? You can create the directory as you what you want. Yesterday I made the new folder because it will take time to like around 1 GB to the massive. So I made it this one. So here is the left panel is the my folder, what we practice. So here's the output folder, what we practice. So low CV and then output there, right? I need the MSTAT input plus one, which will be the quantification result. And then our script, what we practice, it will be good evidence to how we can get it. And then this is the test result, which include the log for changes and so on. And I should upload the metadata about the experiment design. So how I did it is just click it and then just move to the right, and then this is already uploaded there. Yeah, so here's already successfully transferred. And then this MSDAT log file is also the generated from the MSDAT. This also include all the parameter what we use for MSDAT, so it will be good to save it, right? this one actually have a big file, so it will take a time. If you have a raw file, if you want to make a new submission, it will also take a while because it will be like several gigabytes per each raw file. So please have a the good the internet connection. If it's disconnected, maybe you have to start from again. But now it is done. So now all the files, so what I want to submit it is already in the massive space. Then I will go to the web page and then I'm gonna call this one in the using the interface. Then here is the Celebstick data set. Well, so I will sign it. After I sign it, there are several more buttons that show up, right? So before I don't have it, but here's the add reanalysis one and then add file. So add file means not about the reanalysis. At the real top level data set, I want to add the file over there. And then also upload the metadata, which means contact information or keyword and so on. Also add the publication. I already add the publication here. You can also add your publication after your manuscript is published. So here, click the add reanalysis button. Then here you can see workflow, add reanalysis result is selected. And then you can title for that specific reanalysis. So I will say May Institute Sergeant 1 Massive Quant Hands-on, right? There are some limit of number of the characters or required number of the character. We can see if something wrong, they're gonna show up what's wrong. And then the metadata is already selected from our top level data set. So this experiment used the EAST1, used this instrument, and so on. So you don't need to worry about it, but if you want, you can add it as well. Another requirement is we need to add description about this specific reanalysis. So we have to put something here. What is this reanalysis for? So I said, this is the result using the MSD during the May Institute 2020 week 2 hands-on sessions, right? So here we put the title, we put the data, the descriptions, and then here is we need to select the file, which file we want to add for this analysis. So here, for example, peak list and row spectra files already selected because this one automatically selected from containers. So then I want to submit the quantification result, etc. So you can click one of this. So everything link to the one position here. So new window is pop up, right? Then here is the whole list of the data set what I interact with. But bottom, you can see your massive ID. And then click open. And then this one show this right side. This one show you your space, your account space in the massive. Then I made this LFSec folder, right? And then I made a new directory under there, right? And then I submit this one. So I want to submit this file. You can see the files you uploading use the FTP one. There are several files, but you need to tell them which file for which output. So for example, this one, MSDAT input plus the CSV is output of the Skyline report. And then this one is the input for MSDAT, right? Then if you click, you can see this is a highlighted, like light blue, right? If you click again, it's gone, right? So highlighted light blue means select this. I select this and then go up here. This one is quantification result. Then under the quantification result section, this file is selected. And then, okay, the R script, I select it. And then which section? R script is a method. The method section, we can put it in the method section. So this one is now is under the method and protocol sections, right? And then come back again. Annotation is about experiment design. And the metadata, here we have to click this one is some metadata. If you're not sure which file should be which section, so documentation have an example there. So and then show the what kind of information we needed. So you can also check this documentation there. Test result is statistical result, right? Statistical analysis of the quantified analysis. So you can click this one here. And then last, MSDAT and session info, you can just check both of them. And then also should go to the method and then protocol. So we select all of them, right? Yeah, we select all of them. And then the raw file, I didn't select it, but if you submit the new one, select the raw files and then click the raw spectrum file, and then you're gonna submit it here. And then I'm done, the finish selections, right? And then you can see here the quantification result, the one file selected, method, and the 3 files selected, and etc. Right? Then here, next step is you can assign the container. So I want to submit this reanalysis under which container. So here, let's click this. So there are 2 containers there, right? So you can submit one of this, or you can make a new analysis container. So let's make a new analysis container. And then I will say here is the May Institute 2021. And then you can also define the password. And this part, you don't need to worry about it for the analysis part. And then here's show the email me at here. And then if something happened for this workflow, they're gonna send you the notification. This workflow is done or fail, something like that. If you fail, definitely we have to come back and then check it. So then we can just click submit, right? Right, here's the error creating test. This one required upload the peak list is not there. So then, okay, come back. Yeah, and then you can double check this one. Let's say we are not selected this one. Let's remove it, right? And see how it goes. Right. So now massive quant is running to set up the pipeline, the container structure, and then find the location. So it will show up the progress. So it will show the what rows spectra file is linked to this row analysis, and then you can see this. At this moment, you don't need to worry about it. Maybe it will take some minutes, or it's a really big file. It will take some time, but whenever it's done, you can see. Yeah, here's how it show the progress, how it works. Another thing is here in the massive job, it show the workflow what you tried. For example, here click, it show the what I tried. So sometimes I fail, sometimes it's done, and then the last task of what I try. This is the running. So this is the how you can submit the reanalysis one. Then while we are waiting for, let's try to look at the uh, what we already submitted, how it look like. If we succeed, how it should look like, right? So let's go back to the our self-sec data set. Uh, Here we go. So now at the bottom, you can see hold the title and reanalysis container. So I have a question. Do everyone with the account can share new reanalysis? Yes, if this submitting process is successfully done. If fail, it's not, but if successfully done, yes, you can see it. So if you made account, definitely you can see this or workflow. So this one I will show you. So there are 22. I will go back to the very first one. So this is the what I submitted 2 years ago, the same data set with the some setting, right? And then this is the container. So this is the data set, and then this is the container, and under the container, we have the real analysis. So let's click this. So this, I just put a little bit detail description how I analyzed this data set using Skyline. So here's the using Skyline daily version, blah, blah, blah. And then this one is used for this publication and so on, right? And then show this real analysis used 11, have a total 11 files, this gigabyte. Each one have a 6 conditions. And there are around 900 proteins are significantly differently or differently abundant. And then total number of the proteins to quantify is more than 2,000, right? And then if you go to the FTP, you just download this link, you can directly download it. However, there are also some other buttons there. Let's just see how it looks like. So browse the real analysis file. I will click this. So if you click this, it will show the whole the files under this real analysis. So for this real analysis, you can see there are MS, the input is there under the quant directory, right? And then also they have a Skyline G file too. This is it, this is under the others because there's no specific folder for tool specific files, right? So, but you can download this diff file and then you can open it in the Skyline too. And then also R script there to generate this one, and then annotation files, some parameter files as well, and then testing one. So for example, let's click the annotation file here. This is probably smaller. Here's a size 2. Click it, and then you can download it, right? Or here up there, there's a filter by collections, right? So if you click the quant file, it show under the quant file, this file you can see, you can also download it. Quantistat, he also show the quantistat file, which is the output of statistical analysis. You can download it, etc. Quantistat others method have no restriction about the file types. You can upload it any file. So you can upload a PDF, JPG, anything you want. You can upload it this one. This is the how we can browse the real analysis. So I want to go back. Is it still running? Yes. And also, this is already published, and then this is imported from Pride, right? And now this is the public, so it's already public one. But if you want to submit the data set for publication, prefers want to keep it a private before you accept your publication accepted, definitely there's an option. You can just make everything is a private, which means other people cannot see, only you can see that data set. And then also can have you can make ID and password. You can share the reviewers. So, and after everything is going well, and then after done, there's also make a button to make public, And then if you click it, after everything is done, this one will be show up to everybody. So then you can actually share this link. One good thing is if you submit everything is here, in the publication, there are data availability sections, or sometimes you have to fill in some other document. You can give them just this link, and then they can just see all the files under here. We have out of time, so that is the most important part. But I will just show you just other things about the MSDEP workflow there. The concept for MSDEP workflow is currently, let's just come back to our data analysis workflow, right? So we have a raw file over there, and then from the raw file to the final testing output, we still need a several step, right? What we commonly do is we have a public data set there. Then if you want to rerun the identification or quantification, we have to download to the local, right? Download the local and then do the identification or quantification or something. And then we just need to upload all the file up there, right? And then run it. However, now this workflow, if we have a data set available under the here, we can use the pipeline, the workflow in the massive quant, and then automatically save the output under the right the categories. Um, let's go back to our first page in the massive. Here, there's no button to use the MSDEC workflow. Still, just key is to just click the submit your data set, and then we can actually select the MSDEC workflow here. So first, it looked like this one, but if you click here, MSDEC, then give the massive some second. Yes, so now the page look different, right? So we can type the different title so you can recognize it, and then you can select the files, or you can say what kind of tool you use. This is the list of the Spectra data processing tools MSDEC can support. So basically, the MS that have a converter for them or not. So we have a converter for this of this, right? So we can actually use them. So if you want to use the max quant, which means now the massive quant say what kind of input we needed, right? And then the label free or TMT or iTrack. So if you click the TMT, it will gonna call the MSDAT TMT, not the MSDAT, and etc. Then you can also select the files, which is important to run the MS test. Definitely we need the metadata, the annotation files, and then we need the features or proteins. So same as before, click the input. And then you can select your input file here, something like this. Or if you already submit this one under the data set, you can select this button. And then you have to searching your the massive ID. I forgot what is the ID. Let's see here. 8. Okay, let's copy this here and then click filter. Yeah, it will show up, right? And then let's go to reanalysis, and they show the whole reanalysis there, right? And then the hand-on sections. Hand-on. So now you can see this one. Let's say this one is should be annotation, and then I add it. And then the MSDAT, this one should be feature level data, right? And add it and close it, right? So there's a question, what if my experiment type is the SciLec, right? So SciLec, technically there's no MSDAT SciLec one. So we can use the MSDAT TMT for SciLec, but we have to be careful how to make an annotation. So maybe we have to go back to the tutorial what the team covered yesterday. But yes, of course, I like you can use the TMT. Right. So select this. Then you can set up the comparison one. So kind of the one versus the other and etc. So all versus all means all pairwise comparison. Or if you want to click the one means all pairwise against one specific groups. And also you can choose it or you can actually make your own contrast metrics and then you can input it. Here's the advanced filtering. This is the option. Some of the options not cover the whole MS step one. But if you click here, there are some options there. So for example, you want to input yes or no, right? What kind of normalization one? This is the default, but I don't want to, or I want the quantile. If something like this, and then we use the all feature, but I want to use the only top end, or I want to use the high quality. It's based on our feature selection algorithm, and then click submit it. Then same as the what we practice, it will show up this one. Okay, so this one is done, right? So if this is the done, then let's go back to our um, this site, right? So now 22. Let's um, now we have a 23. So here we go. Now we can see our reanalysis submitted in the message quant. Quickly, the other one. So I use the same MStat output. So now every reanalysis have a same format under there. So then actually the message quant to compare our testing result. So this is how we can do it the same as MStat workflow. You can choose the MStat comparison one and then select which analysis, which output you want to compare. If the output format is different, probably have errors. So we should be careful about the output of the testing you want to compare. Last, in our slide, there's a lot of the link for documentation. So please revisit if you want to submit your data and the analysis. And then also there are the videos for each step. So you are stuck for one specific step, please just go there. And then it has more details. So now we have not many times, but it will show all the detail step one by step. So that is all. If you have a question, please let me know.
Thank you so much, Meena, and thank you to everyone who stayed all the way through the end. I think this is a really very important contribution to be able to document all the analysis, not just related to the identification of spectra, but all the way through statistical analysis of quantitative data, and do this in a way that is not specific to a particular tool for data processing or particular statistical analysis software package or anything like that. So even if you are a method developer and develop your own statistical methods or your own data processing methods, this is a repository for you. And also if you are working more on the biology or on the clinical side, and you're interested in documenting your work or just look at the results of the analysis of similar data sets, we also hope that this will be useful for you. So with that, this concludes our session today. However, we will stay back as always in case you would like to ask questions either in the Q&A box, or if you would like to talk to us, please raise your hand and we will promote you to panelists so that you can speak and share screen if needed. And if not, please join us tomorrow. Tomorrow we have a very exciting day. This is the last day of the program. It will have two parts. One is that we offer this platform for contributed presentations for people who develop methods and would like to share their methods with you. We will have 4 presentations by junior investigators. And then the very final presentation of the program is a 2-hour tutorial and a keynote on FragPipe by Alexei Nisvishsky and his group members. So thank you very much. So please don't hesitate to post questions or raise your hand. And otherwise, we will see you tomorrow. Thank you very much. And thank you again to Laurent and to all the teaching assistants who were here for the presentation.